So in this video, I'm going to review the structure of DNA so that we can use it to then more fully describe DNA replication and then look at transcription and translation. So remember that DNA is made up of base units called nucleotides, and it has an anti-parallel double helix arrangement. So I'm just going to start by drawing one side of the DNA and showing the parts. So there are three parts of each nucleotide. There is a sugar, and in this case it's a pentose or a five-sided sugar, and it's deoxyribose sugar, and that's where the DNA part comes from. And then there is a nitrogenous base that's either a purine or a pyrimidine. Purines are either adenine or guanine, and pyrimidines are cytosine, oops, I gotta spell that right, are cytosine and thymine. And when they complementary base pair, one pyrimidine goes with a purine, so A's and T's base pair together and C's and G's base pair together. Now, attached to the sugar is also a phosphate. That's what this is. And the phosphate is the connection that bridges the two different sides, or the two sugars together. So the sequence continues in this way, and we end up with another phosphate, and we end up with another five-sided sugar, and then another in the sequence. So we have got like A, G. Now, the bond between the phosphate and the sugar is a strong covalent bond called a phosphodiester bond. And this bond is difficult to break, which is important because the sequence of nitrogenous bases, the AAC, that's what defines our genes and defines what proteins we make. So we want to easily maintain the order of things, which is why we've got this strong bond, which is called the backbone of the DNA. Now, DNA also has a directionality to it, and it's given its directionality based on where the phosphate is bonding to the sugar. So in organic chemistry, we actually number the carbon rings and give each carbon in this like five-sided sugar ring here a number. And in this case, you have the 3' being right here, carbon, and the 5' carbon being here. So the direction of this DNA is 5' to 3'. So this is the 5', this is the 3', this is the 5'. So you can see the 5 is on the top and the 3 is on the bottom. So that is the direction of that side of the DNA. Now if I were going to draw the other side of the DNA, and I'll draw the little houses upside down just so that you can see the difference clearly. You're going to have, again, this sequence of sugar and phosphate making up the backbone of the DNA, but we're going to see the directionality is different. In this case, you've got the this being the 5', or the 5', and this being the 3'. So you have the 3' on top and the 5' on the bottom. So this side of the DNA is 5' to 3', and then the anti-parallel arrangement, so upside down, the other side would be 3' to 5'. Now, attached to these pentose sugars is also a nitrogenous base that's going to complementary base pair with one of this. So on this side, if I have an A, then on the other side, I am going to have a T. Let's just make the T's boxes, right? Here, again, if I have an A here, that means on the other side, I am going to have a T. Over here, I've got an A, or sorry, I've got a G. So on this side, I'm going to have a cytosine that pairs. Now, they are complementary base pairing, and they are attracted to one another by a weak bond called a hydrogen bond that is easy to break, which is why we can open up the DNA and make copies of it or express genes. And in between the G and C's, there's actually 3 hydrogen bonds, and in between the A's and T's, there are 2 hydrogen bonds. And quite often when we look at DNA replication or expressing of a gene, at the beginning of where this happens, you've got a lot of A's and T's, or it's an area that's rich in A's and T's, because that's a little bit easier to break because there's fewer hydrogen bonds than between the G's and C's. So that is a review of the basic structure of DNA so that we can talk about DNA replication for bacterial chromosomes in the next video.